Hello, chemistry students. Today we will be performing our determination of an equilibrium constant experiment. In this experiment, we will determine the equilibrium constant for the reaction between iron 3 and thiocyanate ions. In part 1, with the assumption that all thiocyanate ions react, we will measure the wavelength that iron 3 thiocyanate complex ion absorbs and develop a standard curve with known concentrations. In part 2, we will run samples with different concentrations of the chemical reaction and compare the amount of products made to our standard curve. Let's get started. To begin part 1 of this experiment, we first need to label 4 50 milliliter volumetric flasks numbered 1 through 4. And we're going to use these 4 50 milliliter volumetric flasks to prepare our 4 standard solutions. And we'll prepare our 4 standard solutions using 0.200 molar iron 3 nitrate in 1 molar nitric acid solution. And we'll use 0.0020 molar potassium thiocyanate. And we'll also be using deionized water. And we'll prepare our 4 standard solutions according to the following table. The temperature of our reagents is 22.7 degrees Celsius. We also need to prepare a blank for our spectrophotometer by filling a cuvette with 1 milliliter of deionized water. We'll now transfer 1 milliliter of each of our standard solutions to a corresponding cuvette. Our standard solutions are now ready to be read in the spectrophotometer. We've powered on our spectrophotometer and allowed the instrument to initialize. So we'll now insert our cuvette with our blank sample, deionized water, into the spectrophotometer and select the Spec 200E Modern Interface option. We'll leave application as live display and go down to measurement mode and set the measurement mode to % transmittance. We'll then set the measurement wavelength to 470 nanometers. We'll move down and select Go, and then we'll blank the instrument by pressing the 000 button. The % transmittance of our blank deionized water sample is now 100%, and the spectrophotometer is now prepared to read the standard solutions that we previously prepared. The % transmittance for standard solution number 1 is 63.0%. The % transmittance for standard solution number 2 is 71.7%. The % transmittance for standard solution number 3 is 80.6%. The % transmittance for standard solution number 4 is 91.1%. You now need to convert your % transmittance readings into absorbance. You can then create a standard curve of absorbance versus concentration for your standard solutions. This concludes part 1 of this experiment. In part 2 of this experiment, we first need to label 3 test tubes, A, B, and C, and then we'll prepare 3 different concentrations of our iron 3 thiocyanate complex ion using 0.0020 molar iron 3 nitrate in 1 molar nitric acid solution And we'll use 0.0020 molar potassium thiocyanate. We'll also use deionized water. And we'll prepare these 3 different concentrations according to the following table. We'll next add 1 milliliter of each of our prepared samples to a cuvette. And we'll now read our samples in the spectrophotometer. The percent transmittance for solution A is 90.3%. The percent transmittance for solution B is 87.1%. The percent transmittance for solution C is 84.0%. This concludes the wet lab portion of this experiment. Okay, students, you can now use your standard curve from part 1 to determine the final equilibrium concentrations of your iron 3 thiocyanate complex ion product in part 2. Remember to convert your percent transmittance readings to absorbance. You can then use your final equilibrium concentrations of your iron 3 thiocyanate complex ion product to determine the final equilibrium concentrations of your reactants. Then plug your information into an ICE table to calculate the value of the equilibrium constant, Kc. This concludes our determination of an equilibrium constant experiment. Thank you for joining me for this laboratory.